Hello everyone, my name is Alejandra Pérez-González. The poster that I'm going to present today is entitled From Genomic Context to Structural Protein-Based Medicine, which is authored by me from the Benemérita Universidad Autónoma de Puebla, by Israel Aguilar Ordóñez from INMEGEN, Mexico, by Norma Caballero from BUAP, and Dr. Enrique Moret from the National Autonomous University of Mexico. This work is part of an exploratory endeavor to describe the genomic landscape of several Mexican Native communities, many of them never before explored by whole genome sequencing. Okay, so previous studies have shown that Native American people display interesting genetic divergence between subgroups, with unique genetic variants and variants that are common among Native Americans, but at the same time rare or even not existent in other populations of the world. In addition, Native Americans have distinctive genetic population structures, which differ greatly from mixed cosmopolitan populations. Such interesting variation was also found in the Comcac, also known as Seri, an indigenous community that inhabits the coast of the state of Sonora in northern Mexico. The Comcac people display long homozygous tracts with more than 10% of the genome in raw, that means runs of homozygosity, and it is also considered one of the most isolated and genetically unique native Mexican communities. The complete genome of four native Comcac individuals were sequenced as part of a study known as 100 GMX that has extended the number of studied native Mexican genomes with whole genome sequencing of individuals from 27 indigenous groups in Mexico. This part of the project aims to identify particular variants for the Comcac group, filter and annotate said variants to find the ones that could create non-synonymous changes in protein coding genes. These particular variants can be defined in our study as those that are present at least 50% of the population studied, in this case the Comcac, present in less than 5% of the rest of the Native American population in the 100 GMX study, and less than 0.5% in the highest population reported in the 1000 Genomes Project. So these are variants that could probably not be studied anywhere in the world. From these variants, we explored and predicted changes in protein structures so we can describe a genomic and proteomic context in a community with interesting features never before studied by whole genome sequencing. To achieve this, whole genome sequences from 4 Comcac individuals were treated. They were pre-annotated and summarized with 2 Nextflow pipelines previously developed in our virtual lab. This produced a BCF file with the discernible variants for the Seri group, which was subsequently split into 3 BCFs, each containing SNPs, indel variants, and other non-SNP variants or indel variants. The BCF with discernible SNPs variants was annotated with assembled BEP and filtered using a script in R. The annotation gave the exact entries for each protein. Protein sequences were extracted from the UniProt database and were modeled in the online platform of SwissProt. The following filtering of the structures was done based on the different features of the computationally proposed models. Communities, values, and other characteristics were analyzed for the different models to choose the best structural prediction for the analysis. So finally, 6 sequences of the reference proteins were obtained from UniProt and used to model the invariant and variant predicted structures. We only found one structure that could maintain major structural changes, a protein corresponding to the canonical transcriptional isoform of the dynein heavy chain domain containing protein 1. Dyneins make up a family of mothers that move toward the minus end of microtubules. Modeled structure presents a serine-leucine mutation at position 1714 of the chain at the upper region of the protein. This position doesn't show non-covalent molecular interactions at the mutation site, and it is expected to be present near to an ATP binding domain. The other model structures for Comcac particular variant proteins have a higher structural confidence, but don't show structural relevance because of various factors. None of the model structures had an exact template elucidated by NMR or X-ray methods, so the structures presented here are only predictions based on similar sequences. In conclusion, the whole genome landscape of the native Comcac people remains largely undescribed, and this community continues to show particular characteristics. It is noteworthy that of the 22 non-synonymous variants particular to this population, at least 6 are related to neuromuscular function. Despite this, most of these mutations studied are predicted to be harmless. Further studies are needed to know if the variants present here generate impacts at the biological and or on the health of the community. Only one of the variance models showed a clear predicted change in structure. This is the first exploratory whole genome analysis of the Comcac community with a structural biology perspective. We hope that this study sparks more interest in exploring in silico implication of structural biology based on medicine. Thank you for your attention. Any question can be answered by my email. Have a nice day and don't forget to check the other topics in this Congress.